In this video, I'm going to explain the setup for the germination investigation and describe how you're going to make a data table to collect the information from the investigation. So, as you can imagine, in this investigation, we're going to be looking at the germination of some seeds. We are going to be using pea seeds as our sample organism. The question we're going to be asking with the pea seeds is, how does temperature affect the rate of germination in those pea seeds? And we're going to be looking at the actual germinating seeds and non-germinating seeds as well. When we talk about the rate of respiration, this is a process that all living organisms go through in order to make energy, ATP, for themselves. So respiration is something that happens in all living things, but the first question is, will it be happening in the non-germinating seeds as well as the germinating seeds? And the second part of the question is, does temperature affect that, how quickly that's happening? So here's the overall equation for respiration. You don't need to be overwhelmed by this because I guarantee you recognize all of the molecules here. So this first molecule, C6H12O6, is our glucose. That is the fuel for respiration. And the next molecule is oxygen. Okay, so all living things need glucose and oxygen. Plants, animals, bacteria, everything relies on those two things in order to make, down here at the bottom, this is the second part of the equation, so this is some product, our energy. Remember, that was the ATP I was talking about. And then as a byproduct, carbon dioxide and water. So these are waste products given off by this cellular reaction or cellular respiration reaction. We know exactly how much energy is released. It's 686 kilocalories of energy from starting with 1 mole or 1 unit of glucose molecules. The reason there are some numbers or coefficients in front of these molecules is this is a balanced reaction the way it's written. So with that in mind, the question down below says, how could the rate of respiration then be measured? How could we figure out how fast respiration is happening? Well, we could calculate how much glucose is being used, so how fast the glucose is being used up, or how fast the oxygen is being used up. Or we could calculate how quickly the CO2 is being produced, how quickly the water is being produced, or just how quickly the ATP is being produced. For our purposes, the easiest thing for us to do in class with the materials we have is to record or to follow the consumption or the use of the oxygen gas. Okay, so the faster that oxygen gas is being used, the more respiration is taking place, the more ATP is being produced. So that's what we'll be measuring in this investigation. Now, how do we know how fast oxygen gas is being used when oxygen gas is a colorless, pretty much invisible gas floating around in the atmosphere that we don't usually even see? Well, we're going to use actually a physics equation to figure out how much oxygen gas is being used. Okay, so the way that we're going to measure how much oxygen is actually used in this reaction is through this equation called the general gas law. So in order to understand that, we're going to step through some of these variables. You can see the names of all the letters, what they stand for below. We've got pressure, volume, how many molecules of gas, and the temperature of gas. The R is the gas constant. That's a number that never changes. So we're always going to kind of leave that out of our discussion because what we're interested in is how the reaction or how the equation does change. Okay, so we're going to disregard that constant for now. So we're going to go through some of these questions over on the right-hand side to help you understand the general gas law a little bit better and then eventually apply it to our own experiment. So if we keep in a system of a gas, so imagine you've got some gases here in a container, okay? If we keep the pressure and the temperature on that container constant, So I'm going to just cross out the pressure and the temperature and assume those don't change. What happens with the volume and the number of molecules? Well, the gas law says if we increase the number of molecules, so we have more molecules in our system, the volume will have to increase because the two sides have to stay equal. So the size of our container will expand. And the opposite happens as well. If we decrease the number of gas molecules in that container, the volume will decrease. Okay, so hopefully that makes sense. That's one of the things that the equation can tell us. Now let's look at the next question. What if we keep the volume and the temperature constant? So again, we're going to ignore this R value right now, and we're going to cross out the value or the volume and the temperature. What happens with the number of molecules and the pressure? If we increase the number of molecules, the pressure will have to increase. If we decrease the number of molecules, the pressure will have to decrease. Hopefully that makes sense. Next one, what if the number of molecules, the N, and the temperature are constant? So we're getting rid of everything on this side of the equation. Well, lots of different things can happen here. So if we increase the pressure on the container, the volume on the container is going to decrease. Okay, so here's our container, and we put some pressure on that container. Think about like squishing a can. The volume is going to decrease on that container. Okay. If we do the opposite, if we decrease the pressure, then the volume is going to increase. So the container is going to get a little bit bigger because those gas molecules aren't quite as packed in. All right, the last one is going to be regarding the temperature and the number of molecules. So if we, again, cross out that constant and the number of molecules is constant, what if our temperature changes? So if our temperature increases, then the combined effect of the volume and the pressure has to increase. Okay, so together those two both have to create an increase. If the temperature decreases, again, the combined volume of those has to decrease as well. And this one's going to be kind of important in our experiment that we do, because we are going to be increasing and decreasing the temperature, and as a result, watching the increase or decrease in the pressure and volume of our gas. So the volume is going to be the volume of oxygen, and we're going to be able to relate the pressure of the oxygen to that increase or decrease in the volume. All right, so keeping this in mind, what we are doing in our investigation is, again, we're changing the temperature. So that's our independent variable. Remember, the I in independent is for what I change in the investigation. So we're going to do a cold temperature trial and a warm temperature trial. What we are actually looking for, the result of changing that pressure or changing that temperature, is going to be a change in pressure in the system. So we're going to see if the temperature change changes the pressure. And why would we look for a change in pressure as a measurement of oxygen? Because remember, the whole point of this is to measure how much oxygen is consumed. But remember, according to our gas law, if we change the volume of a gas, in this case our volume of our oxygen, so here's our volume, it will also change our pressure. Okay, so if again the volume of a gas goes up, the pressure of that gas is going to go up. And the volume of the gas goes down, the pressure of that gas is going to go down. Okay, so we can tell how much oxygen is being produced based on the actual pressure change in our system. Okay, so the pressure and the volume are related. Okay, how we're actually going to measure this, like the actual equipment we're going to use, looks like these guys. These are called respirometers, just a fancy name for how to measure respiration. And I'm just going to quickly take you through the stuff that's in them. So at the very bottom, we're going to put some cotton with potassium hydroxide soaked into that cotton. And I'll explain in a minute why that is. And then on top of that soaked cotton, we're going to put some more cotton that can't absorb the potassium hydroxide, because we want to keep the potassium hydroxide on its own down there. So the non-absorbent cotton is going to create like a barrier. On top of that non-absorbent cotton is going to be, in one of the respirometers, some germinating peas. So peas that have already started to sprout, essentially. So that's number one. Respirometer number two is going to have the same thing with its absorbent cotton and potassium hydroxide at the bottom, non-absorbent cotton next, some glass beads on top of that, and our dry or non-germinating peas, because remember we wanted to see germinating versus non-germinating peas. The reason, however, that we add these glass beads is because we want the volume. See how they're kind of at the same volume here? We want the volume to be the same between our germinating and non-germinating peas, because remember, volume is an important variable in our gas law equation. So we want to keep that constant as far as the actual contents of the respirometer goes, so that any changes in volume are due to the volume of the gases that are produced, not the volume of the peas. Okay, so this is kind of our, experiment right here is our dry peas versus our germinating, non-germinating versus germinating. But then we also need a control. So that's respirometer number 3. Again, at the bottom, our cotton soaked in potassium hydroxide. On top of that, the non-absorbent cotton. And then on top of that, all beads, no seeds at all. But again, look, the volume is going to be the same. Okay, so the volume of the solid materials in all 3 respirometers will be identical. Then the last thing that's going to happen is you're going to set up a set of 3 of these again. So in the end, you'll have 6. You'll have 2 1s, 2 2s, and 2 3s, because the other variable you're testing is cold versus warm. So some of these will be in a warm environment. Let's say these 3 are in a warm environment, and an identical set of 3 will also be in a cold environment to measure the respiration. Those environments are going to be dunked into a tank of water, so cold water or warm water. And as the cold or warm water warms up the peas, respiration is going to happen and the pressure is going to change. As the pressure changes, the water is going to go into these little tubes, little pipettes that are stuck in the top of the respirometers. And the greater the change in pressure, the more water will be soaked into those tubes. So that's how we're going to measure the pressure change. And remember, the pressure change is a direct relation to the change in volume of the gases inside that respirometer. Now remember, we wanted to measure oxygen gas. And oxygen gas isn't the only gas, though, that's in that respirometer. Remember, oxygen gas is being used, but carbon dioxide gas is also being produced in this reaction. So how do we get rid of the carbon dioxide gas? How do we make sure that that carbon dioxide gas isn't contributing to the pressure change? Well, in this reaction, we've got some potassium hydroxide that we've added, and potassium hydroxide will actually bind to carbon dioxide to create water and a solid. Potassium carbonate is a solid. So if we take out that CO2, any change in the volume of the gases inside our respirometer will be directly related to changes in oxygen, because we've essentially eliminated the effect of the carbon dioxide on the pressure change. All right, so to sum it up again, what you're going to have is essentially 6 respirometers. 3 of them will be at room temperature with the germinating seeds, the dry seeds, and then the control, the beads. And then 3 of them will be at a colder temperature with the germinating seeds, the dry seeds, and the beads. So those are our warm and cold temperatures, and then our germinating versus non-germinating in both temperatures. All right, your job to prepare for this lab for tomorrow is to get a data table set up. You're going to be collecting data on all 6 respirometers, and that data, remember, is going to be based on that pipette, how much water flows into that pipette. So you're going to be recording data in milliliters. You're going to be checking the volume in milliliters of each of these respirometers every 5 minutes for 20 minutes. So you're going to have a 0-minute reading, a 5-minute reading, a 10-minute reading, and a 20-minute reading for each of the respirometers. And again, the readings you're going to be collecting are in milliliters. So I'd like you to have a data table ready to go on your iPad so that you can put that information in for all 6 respirometers as you collect the data tomorrow and the following day. Remember, send me any messages you have on Schoology and I'd be more than happy to help you out.